Thank you for joining us for our AMS May virtual seminar. Before we begin, I'd like to acknowledge the traditional owners on the land on which we are all meeting today. I'm speaking to you from Meanjin, Brisbane in Queensland. This is the traditional homelands of the Turrbal and Yagara peoples, and it stretches from the North Pine River across the Brisbane River down to the Logan River. I pay my respects to the elders past, present and emerging. Our presenter today is Dr. Sarah Sapsford. Sarah recently started a new position as a senior advisor in plant risk analysis for biosecurity New Zealand at the Ministry for Primary Industries. Originally from British Columbia in Canada, Sarah moved to Queensland, Australia in 2009 to complete her Master of Science at James Cook University. During this time, she studied the amphibian chytrid fungus and has been responsible for many, declines and extinctions around the world. She then moved to Perth, where she completed her PhD at Murdoch University in forest ecology and pathology. This project introduced her to the vast species of fungi found in forest soils. After the completion of her PhD in 2018, she took on a postdoctoral research fellowship at the University of Canterbury in Christchurch, looking at how invasive plants and their symbiotic fungi alter native grassland ecosystems. Today, Sarah will be talking about her work by presenting 2 case studies on how fungi can change ecosystems and how these fungi interact with other members of their community. If you'd like to ask a question or post a comment for Sarah, you can do so by clicking on the Q&A button at the bottom of your screen. Sarah will answer your questions at the end of her presentation, or you can put your hand up and talk to her directly at the end of her talk. Then, after the question and answer session, at the end of the seminar, we will take a moment, if you wouldn't mind taking a moment to answer a short survey just to help us improve the delivery of this seminar series. So thank you, Sarah. You can take it away.
Thank you very much. I'm just going to share my screen. There we go. So thank you very much for inviting me to speak today. And as Tracey said, I'm going to be presenting 2 case studies on how I believe fungi can change ecosystems. But first of all, I just wanted to give a little background on fungi around the world. And so to date, there are approximately or almost 150,000 species of fungi that have been identified and described. And it's actually predicted that this only makes up 10% of fungi on Earth. Now, there's a bit of debate on this number, as some literature says there's between 2 million to a possible 15 million species of fungi on Earth. And this is quite possible because if you look at the pictures I've presented here, these are fungi that are actually visible to the naked eye. But there are so many fungi that you can't see. And what I wanted to talk about first is the role of fungi in ecosystems. But before we go into that, we first have to understand what is an ecosystem. So I'd like you to take a moment and picture in your mind what you think an ecosystem is. So just take a moment. And is the first thing you pictured maybe the ocean? Or maybe it's rivers and streams. Or you're probably more like me and your first thought goes to forests. Well, what is an ecosystem? According to the dictionary, it's a biological community of interacting organisms and their physical environment. And this, of course, includes fungi. So when you pictured your ecosystem in your mind, did you think about the role of fungi in those places? What's really interesting is fungi are found everywhere. So in marine environments, in terrestrial environments, and rivers and freshwater systems. But for the purpose of today's talk, I want to focus on terrestrial environments and particularly forests. And in forests, fungi have quite a diverse role. And this is a nice overview from a publication a few years ago. If you have a look at the top left of the screen, you can see that fungi in forests have supporting roles. And here they provide habitats and food for many animals, as well as improving soil structure and soil fertility. On the top right, we see that they have regulating roles where they help sequester carbon. They control the diversity of plant species by acting as pathogens, but then there are some that also protect against those pathogens. If we have a look at the bottom left, we can see that there are provisioning roles where they help with tree biomass. They also help with crop and fruit productivity. They're food for us, as well as in some cases, they have medicinal properties as well. And one of my favorite roles is that bottom right panel, and you can see that they have significance in our culture, specifically in art. And for many of us, we like to go out into the forest or any other environment and have a look at the fungi and take photos. Well, that's what I like to do anyways. So keeping these roles in mind, I'd like to first discuss my first example. And this involves this forest tree species, which is endemic to the southwest of Western Australia. It's Corymbia calophylla, or the common name Mary. And this was the topic of my PhD thesis. You can see that they produce these wonderful white flowers and these fruits called honky nuts. Now, Mary is an important species as it's a food source for many birds, especially parrots, as well as nesting locations for these birds, including the endangered Carnaby's black cockatoo. And they're also an important tree species for the honey industry. So overall, this species provides a lot of benefits to a variety of organisms. However, in the 1940s, people started noticing these cankers on Mary. But at the time, they weren't of serious concern because only a few individuals were affected. But by the 1990s, almost 50 years later, these cankers started causing noticeable decline of this species. And this is a great photo from a colleague of mine, Trudy Papp, which shows a time lapse of a single tree over 13 years. So if you look on your far left, you see the tree has quite a severe canker. And then 13 years later, that tree has succumbed to that canker disease. Now, these cankers are caused by the fungus Quambilaria corycup, which essentially girdles its host or strangles it in a sense. And trees try to defend themselves by walling off the infected area. So you can kind of see it's tried to do that here on the left-hand side. But this pathogen essentially penetrates this wall and reinvades and causes the canker to get bigger and causes the death of the tree. Now, it doesn't only infect trunks of trees, as you can see on the left here, but also in branches. On the right, you can see some quite severe cankers on branches of these trees. One of my favorite photos from Trudy is this one, and this is a canker just starting, but this white patch here is the fungus sporulating, essentially reproducing and spreading its spores probably through wind dispersal. We have been able to culture this fungus, as you can see here. We've been able to grow it on different types of media, and this is what it looks like under a microscope on the right-hand side. Now, interestingly, this fungus has not been observed outside of Western Australia, and so is thought to be endemic to WA. And what's unusual about endemic pathogens is they don't usually cause declines. Most of the time we hear about, invasive pathogens that have been introduced to an area, and they're the ones that wipe out species. However, if a tree becomes stressed, or to a point that it's so stressed it can no longer fight off disease, so it becomes susceptible to these pathogens. Many of you have probably experienced this, like myself. If you get stressed and you get to a certain level of stress, you're more likely to catch a cold, and you're not able to fight that off properly. And one of the biggest stressors for Mary is habitat fragmentation and land use change. And when I say land use change, I just mean that area was probably once a forest and it's been changed to a different type of land use, such as agriculture. And so specifically in Western Australia and other parts of the world, extensive land clearing has led to a highly fragmented landscape, as you can see on the far right up here. And what happens is forests become situated in a matrix of different land uses. So that includes urban areas, agricultural land, land for grazing, for example. And what's so unusual or interesting about these fragments is that the edges of these forest fragments experience different environmental conditions, including greater fluctuation in temperature, in moisture. They're exposed to wind and different rates of evaporation. And the exposure to these edges depend also on the type of land use they're adjacent to. So for example, there might be stresses from compaction or irrigation. And if you're close to agricultural land, there could be potential herbicide and nutrient runoff. Now, many trees are quite sensitive to these changes. So along those edges, you might also see a loss in tree or plant diversity. In addition, these edges provide greater access by people, by us, as well as fire and invasive species, such as other pathogens that could be introduced to those areas. Now, historically, Mary is actually quite tolerant to stresses in comparison to other species, such as like Jarrah in that same area. And it has managed to survive over the years, obviously up until now, and it's become susceptible to this canker disease. And one of the things we've observed in this fragmented landscape is that we see a higher canker incidence along disturbed edges. And what I mean by disturbed edges, if I just ask you to focus on the picture on the right, is what happens is we have these highly fragmented disturbed edges where there's remnant stands of Mary, and they're adjacent to two types of land use. So for example, in this picture on the left, you have some sort of agricultural land, whereas on the right, you have a road. On the opposite side of the road, you have what we've called a forest edge, which is only adjacent to one type of land use, which is the road. And then 200 meters in, you have the interior of the forest, which is not experiencing any type of edge effect. And on the left here in this figure, we can see that canker incidence here. So we have proportion of canker trees on the y-axis and the different edges on the x-axis. And so along those disturbed edges, we see up to, or on average, 45% of trees have cankers. That drops to almost half, to 20% on the forest edge, and then almost down to 0 within the forest, but we're still seeing a little bit of canker within the forest. However, I've talked a lot about this pathogenic fungus, which is destroying these trees, but there are also other fungi involved that are actually beneficial for trees. And many of you probably know mycorrhizal fungi are beneficial soil fungi that associate with Mary, but as well as many other plant species around the world. And essentially they infect plant roots or colonize plant roots and spread through the soil, increasing the surface area through which plants are able to get nutrients. They also provide, in some cases, resistance to root pathogens, and they can also improve soil structure. Now, there are two groups or two types of mycorrhizal fungi. For example, the one pictured here, what they do is they produce a covering over the fine roots or a mantle, spread through the soil, and they can produce these fruiting bodies. The other type of fungi are arbuscular mycorrhizal fungi, and these also penetrate root tips and spread through the soil, but they don't produce fruiting bodies. Both groups essentially help plants survive. They increase the health of the plant. And many of you have probably seen experiments where they plant trees or plant different species with and without mycorrhizal fungi, and those with mycorrhizal fungi tend to grow bigger and look healthier. But just like their hosts, their trees above ground, these fungi are highly sensitive to changes in their soil microclimate. So changes in moisture and temperature, the amount of soil nutrients. And so they're most likely sensitive to habitat fragmentation and land use change, because we're seeing these differences or exposure to different things on these edges. So this was actually the main question for my PhD, is I wanted to understand how do mycorrhizal fungi respond to edge habitats, and particularly different types of land use. So what I did was I sampled sites throughout the Southwest, and you can see here on the map, all those red dots are the sites that I went to, and at each site I had those three types of disturbances. So on that inset there, we had that disturbed edge, which was essentially adjacent to those two types of land use. On the opposite side of the road, we had the forest edge, and then we had the interior of the forest, which is experiencing no effects of land use change. And so what I went out to do is I collected soil from all of these sites, and the first thing I asked was, how is disturbance or this habitat fragmentation changing the soil? And mainly soil nutrients, as well as properties such as moisture and temperature. And from there, I looked at the communities of mycorrhizal fungi associated with Mary at each of these sites. So one of the first things I found was we saw an increase in soil nutrients along that disturbed edge. So I don't want you to focus on this whole table, but if we look at the main nutrients such as nitrogen, we see values are higher along that disturbed edge in comparison to that forest edge, and then even more so to the forest. And this was a similar trend for phosphorus as well as potassium. And it wasn't just the nutrients that changed, it was also other properties such as soil moisture. So interestingly, both edges, the disturbed edge and the forest edge, had much higher soil moisture in comparison to within the forest. And one interesting thing we also noticed was leaf litter depth along that disturbed edge, as well as the forest edge, was much higher than within the forest. And that's something I want to come back to. So one thing we think is happening is these disturbed edges are possibly getting runoff or nutrient runoff from agricultural land, which is increasing those levels of nutrients. And you'd think this could be a good thing because more nutrients is better for plants, but they do have an optimal range where it could potentially be above optimal, so it could be toxic, or even below optimal, where they're not getting enough. And unfortunately, we don't have that range for Mary. That's an area that needs more work. But in either case, we're seeing higher levels along those edges. So now that we're seeing these changes in the soil conditions, what are actually happening to the mycorrhizal fungi? What we're seeing is a decreased diversity of mycorrhizal fungi. So on the left graph, we have diversity on the y-axis and those transects, or that different edges on the x-axis. And we see that on average, there's higher diversity within the forest in comparison to those edges. And what was interesting is the disturbed edge and the forest edge, there was no significant difference. So it didn't matter if you were being affected by two types of land use versus one type of land use. So what I did is I wanted to take all of this information and put it in a model to determine main drivers or main influences on canker incidence in these areas. And so putting that all together, we found that canker incidence was highly correlated with leaf litter depth and diversity of mycorrhizal fungi. So if I take a look on the left, we have the proportion of trees with canker on the y-axis, and we have leaf litter depth on the x-axis, essentially showing the deeper the leaf litter, the more likely trees will have canker. And we see the opposite trend with mycorrhizal fungal diversity. As diversity increases, canker decreases. Now, these are obviously, at this time, only correlations. We need to do more mechanistic experiments to figure out cause and effect. But I do have a working hypothesis, which I've observed over my time, which I think is happening. And so essentially along those disturbed edges, we're getting higher canker incidence, and that's for multiple reasons. But with more cankers, you're getting more tree death, branch death, and that causes more leaves to fall to the ground. So you're getting an accumulation of leaf litter. And this is probably at a higher rate than it can be decomposed. And essentially this layer of leaf litter, it has an insulating effect on the soil. So we see higher moisture levels in those areas in comparison to within the forest. And these changes in microclimate could essentially be driving the decrease in mycorrhizal diversity. And because mycorrhizal fungi are so important for tree health, indirectly, they're affecting the health of the tree, which is making them more susceptible to this disease. So it seems like a positive feedback loop that is occurring. So just to conclude the first case study, what we realize is that this is quite a complex decline of Mary. There are a lot of factors involved, but essentially in the face of global change, endemic pathogens may become more prevalent, and we could potentially see this in other parts of the world. And part of that is because of habitat fragmentation and land use change. These two things are altering soil properties as well as the soil fungal communities, which indirectly are affecting the health of these trees. So now I want to jump to quite a different topic. So I focused here on, pathogenic and beneficial fungi in a forest ecosystem. And now I want to switch gears and talk about co-invasion of pine and ectomycorrhizal fungi. And for this example, I'm going to focus mainly on invasions in New Zealand, as this is where I did most of my work. So pines have been introduced globally for timber and pulp, amenity values, and erosion control. And these introductions have led to widespread invasions, especially in the Southern Hemisphere. This leads to huge ecological and economic problems. We've seen this in New Zealand, in South Africa, Argentina, and Chile. For example, New Zealand spends millions of dollars every year trying to get rid of these trees where they've invaded into the wrong area. But what was really interesting is when they first brought over pines to New Zealand, they couldn't survive and they couldn't establish. And that's because pines have a very close association with ectomycorrhizal fungi. And these are the fungi that produce that mantle and these fruiting bodies. So 2 fungi you may have seen near pines include Amanita muscaria on the left here and Suillus luteus on the right. And so essentially what happened was they started bringing over the fungi and planting pines with the fungi so they could establish. And that's what's happened to produce forestry industry here. Now, this becomes a problem when pines escape those plantings, usually by wind dispersal through seed. Fungi are also dispersed by wind, and in this case, animals also disperse. So essentially, these fungi get brought into these new areas and trees are able to establish. And we might ask, oh, well, what's the problem? We want to start planting more trees to help with carbon emissions and everything. But this essentially comes down to the wrong tree in the wrong place. Pines aren't native to New Zealand, and neither are their fungal partners. And so we don't specifically know what the impacts of this association is. One thing we do know specifically in New Zealand is they do cause obvious changes above ground. So here they invade into grasslands. So as pines invade into grasslands, they produce monodominant closed canopy stands. So on the left here, you can see a grass shrubland in New Zealand. There's a lot of plant diversity, different grass species, shrub species. And in the background, there's actually a native beech forest. In comparison, on the right, we have a established pine stand. There's no other plant species around, and it's just pine. So therefore, as these pines invade, there's a loss of plant diversity as pines displace other species. And this process is known as biotic homogenization. Essentially, these invaded plants homogenize the environment. However, one question we have is, what are the ectomycorrhizal fungi doing below ground? So specifically, these types of fungi aren't found in grasslands. They have different groups of fungi associated with them. And what we see, the high plant diversity in grasslands produces patches of different grass and shrub species, which may create diverse soil conditions and thus support a diverse fungal community. In comparison, pines only associate with a few of those ectomycorrhizal fungal species. So would we see a decrease in diversity with the introduction of those fungi, or maybe an increase because we're introducing new species? So that was the main question, and this was during my postdoc. We wanted to know, how do pine and their ectomycorrhizal fungi alter soil fungal communities? So we went out to a site just outside of Christchurch where a shelter belt had been planted and pines had started to invade into the grassland behind that shelter belt. And we sampled soils from across a pine density gradient. So where there were no pines in the grasslands, as well as on the right panel, you can see low pine density, medium pine density, and then high pine density. And so we wanted to see what happened to the fungal community across that density gradient, essentially. And what we found was actually quite surprising. We saw that pine invasion causes a loss of fungal diversity. So in this figure, we have diversity on the left, the y-axis, and we have pine density on the x-axis. And we actually saw a loss, a fungal diversity decreased by 50%, which seems huge. So that 0 here indicates grasslands, and then the 120 is the high density pine stand. This is in basal area. And this one graph shows you the entire fungal community, and it hasn't been separated into the different groups of fungi. So we were also interested in what is happening with ectomycorrhizal fungi. Interestingly, we see no change in diversity of ectomycorrhizal fungi. And we were like, okay, well, this is interesting. What about other groups of fungi? Well, we focused also on saprotrophs, and those are your decomposers. You usually find them on decaying wood or in leaf litter, decomposing soil organic matter. And when we looked at the diversity of that group of fungi, we see that it mimics the trend of the overall fungal community. And so this was quite interesting, first of all, because we saw ectomycorrhizal fungi were already present in the grassland. So they're already there, which means that pines can essentially come in and establish successfully. And overall, this shows, once again, biotic homogenization. These fungi are homogenizing the below ground community. But we wanted to understand this relationship between the saprotrophs and the ectomycorrhizal fungi and what specifically is happening. And so one thing I was going to briefly discuss is the role of fungi in soil nutrient cycling. So fungi play important roles to break down nutrients so plants can take them up. And grasslands and forests tend to cycle these two things differently. On the left, in grasslands, you can see soil organic matter is broken down by the soil biota. This includes bacteria, your saprotrophs, and other soil fauna. And these produce extracellular enzymes, which are able to break down that soil organic matter, produce mineral nitrogen that is easily taken up by plants. The same thing happens in pines or forests, except for two main differences. There is an introduction of different litter quality and quantity. So these are your pine needles, and they're actually really difficult to break down. And so this change in chemistry essentially changes that community of decomposers, and thus changes the activity of extracellular enzymes. The second difference is these ectomycorrhizal fungi also have extracellular enzymes, and they're also able to break down soil organic matter, providing nutrients for their host. So what we think could be happening here are saprotrophs that are part of the soil biota, and the ectomycorrhizal fungi are competing for the same resource, so for the same organic matter. And essentially, ectomycorrhizal fungi are winning. They're displacing those saprotrophs. But once again, we haven't mechanistically tested this. So overall, for this case study, the co-invasion of pine and their ectomycorrhizal fungi are causing biotic homogenation above and below ground. The picture on this slide shows a relatively native grass shrubland in New Zealand. And so we're essentially displacing those species in those systems. And because of these changes in nutrient cycling, we're seeing changes in the nutrient pools in the soil, as well as changes in pH. And this can have effects on future plant establishment. Some plants won't be able to survive in these new conditions. And so essentially we could be inhibiting recovery and restoration of these co-invaded systems. So that is everything I have to talk to you today. I hope I've shed some light on how fungi can change ecosystems. And I just wanted to thank and acknowledge all my colleagues and collaborators who have made all this work possible over the past few years. So thank you very much.
Thank you so much, Sarah. That was really interesting. Great to see a couple of different case studies there, highly relevant to the Australasian Mycological Society. So thank you for touching on both Australia and New Zealand. I'm going to start with questions relating to your first case study. So Philippe from Peru, welcome. He asks, what was the protocol that you used to measure mycorrhizal diversity in your first case study?
Okay, so we first, we obviously collected soil. We did DNA extractions on that soil. And then we sequenced the DNA using Illumina. And we used primers. If you're interested in primers, FITS7 and ITS4. And essentially from there, we've developed a pipeline to essentially cluster those sequences and blast them against the database to figure out species. And then from there, we looked at Shannon diversity specifically. Please let me know if I've answered your question or if you'd like more details.
That sounds great. Thank you. So was that a similar process? Did you also use Illumina for the New Zealand study?
Yes, so the same primers, similar pipeline, similar methodology there as well.
Okay. So I have a question about that study. First of all, what is a shelter belt?
So I don't know if that's
familiar with.
I don't know if that's like a New Zealand term, because I also didn't really know. But properties, I mean, so Canterbury is quite flat. And so to protect, I guess, your house or your land, you plant trees that kind of protect you from the wind. So it's just some kind of barrier, in a sense. And people really like pines, and I have absolutely nothing against pines because in Canada they're all over the place.
I do love the smell of hot pine needles.
Exactly.
I can get it. Yeah. Shame that they've decided to become invasive now too. So part of the study that for the grassland diversity, you found that Ectomycorrhizal diversity didn't change, but I wanted to know, what about species composition? Did you find more of certain types of species than previously when they were grasslands or in grasslands?
Yeah, so I didn't put that in the talks. I think I ran out of time. But yeah, we did see a change in composition. So what's really interesting is the composition in grasslands was completely different to, like, let's say, dense pine areas. But there was this intermediate, I guess, composition where there's still grassland species present and pines are kind of coming in, kind of medium density, where the community composition overlapped the two extremes and then just tipped over. But we also saw changes in, I guess, relative abundance of certain species. So those that are really important for pines, so like Amanita and the Swillis I showed photos of, they increased their abundance as pine density increased. So we lost some species, but gained some new ones. And the similar thing happened with saprotrophs as well. That whole community shifted.
So then what are your perspectives on sort of restoration? So if pine stands had to be reduced in size or eradicated, do you think that it would be essential to then alter or restore the soil health by essentially inoculating with mycorrhizal species from the desired ecosystem?
That's a really good question. Part of my work that I'm still working on is looking at legacy effects of pine. So if we remove pine and we leave the soil, what happens? Can it be restored on its own over time? And preliminary results from this work suggest that there's a possibility, but it could take years. So, and I've only looked up to 15 years. So even after 15 years after pine's been removed and they stay removed from that area, soils still aren't the same as they were, 15 years previously. But it is a possibility. And this just also comes down to understanding native fungi in New Zealand. One of the things I wanted to answer was, are we losing native saprotroph fungi in the soil? But it's really difficult to figure out what are native, what aren't, because we don't have that information. But once we do have that information, I think it is possible we could do something like that.
Yeah, really interesting. I think it's an issue that's going to be more researched here in Australia as well with our dieback syndromes. And I think we've got another question coming through that Philippe also says thanks for answering his previous question. The tree is only found in Australia. I assume he's referring to Mary.
Yes, so this is only found in Western Australia. That specific, that specific species. I'm not sure if if people take it and plant it elsewhere.
Yeah. Okay, but it is endemic anyway to WA.
Yeah.
Awesome. If there are any other questions, you're welcome to raise your hand or to type it into the Q&A section. Sarah, thank you so much for your talk today. Really enjoyed it. And good luck with your new role at Biosecurity New Zealand. We'll look forward to seeing what other work you do over there. And yeah, thank you.